So I think really sort of for this first session, we wanted to discuss sort of setting the stage and why. And some of our early preparatory work, we were thinking about, you know, every field sort of has this question of why things perhaps stop working. And in brain science, the moment came when we realized that despite all of our data and our tools, we still don't understand some fundamental aspects. And that one question would be why some neurons stay alive and healthy for 100 years and while others perhaps failed decades earlier. And so I think maybe this is where we'd like to start is what's the biggest unanswered question about the brain that still keeps you awake at night? And we will have hopefully some interesting discussions about this. And to sort of maybe set the stage, I'd like to maybe just start with Olivier, since you're seated next to me. You know, we've been discussing this topic in terms of where some of the challenges and opportunities are, and certainly in terms of collaborating with everybody within CLARA, but also in other areas. The amount of information that we've generated and we continue to generate, what are some of the impediments to really sort of being able to share data and to gain insights that you've run into?
Oh, sorry. Thank you. Yeah, also from my point of view, well, there are so many important questions, but what fascinates me, I think, the most is how fuzzy the boundaries between different brain disorders are. And I think it was very well highlighted in one of the talks, but it goes even further because it goes beyond neurodegeneration. It's actually across all the span of brain disorders. And I think that's one of the main barrier to finding effective treatments. So in a way, we need to have a sort of redefinition of what these disorders are and what are their boundaries. And so that's the first start.
That's terrific. Thank you. You know, in terms of sort of identifying these boundaries, you know, something as a statistician, and I've often heard this from people in the public, you know, they will say, well, you've been doing research for a very long time. Why don't you just go collect a bunch of data, put it all together, and then just start using it? And it's obviously that's sort of a lay interpretation of maybe perhaps what we do in research, but what are some of the practical limitations in really trying to do that? What can we tell the public in terms of, you know, really trying to do this type of research that we, you know, need to sort of be very careful about the types of data that we collect? And I think in Adam's presentation, he talked about heterogeneity. How do you sort of see data being collected and really being developed across the globe, and how easy is it really to use it in an interoperable way?
Well, maybe at least I can speak about one aspect, which is I think one of the big challenges is to be able to connect the highly precise, very sophisticated data that we collect in research to that collected in clinical routine that we have in hospital data warehouse. And so that's really one of the big changes we've seen in the past years, where almost all hospitals in the world have created these repositories where you can access clinical routine care. Actually, even more than that, for example, in France, you can access reimbursement data, so you have a kind of trajectory of the entire patient throughout their life. So this data is rich by its breadth and also its time frame, but at the same time, it's very narrow because it doesn't give you a precise a view of the molecular aspects or, for example, the detailed imaging or this kind of thing. So I think one of the biggest practical bottleneck we have is how do we bridge this together to get the full view of the patient.
No, that's actually a really great transition to Stefan. Your background is really running supercomputing centers and managing large compute centers as well. Where do you see sort of the greatest potential for high performance computing to sort of deal with this translational problem that we have to deal with between research and real life medicine and real world evidence?
Well, I think first of all, the potential that we have in the supercomputing centers, and CLARA is supported by 2 national supercomputing centers, by IT for Innovations in Czech Republic and by Leibniz Rechenzentrum from Germany, which is in Germany one of the 3 centers that host national supercomputing. It's the evolution that we are just undergoing from pure classical supercomputing to AI and quantum computing and combining all these systems together. And that's exactly the idea, that CLARA is not just based on classical simulations with high performance and with really world-class supercomputing, but also mostly by AI and then with the support of the really novel quantum computing techniques. And I think there we are building lots of experience and we are delivering infrastructure that will be really a booster for the center. Yeah. Beyond that, there's a data thing. Maybe I would like to comment on this data collection part, because of course this data collection part has a semantic aspect, but then there is the aspect of bringing the data really together in order to deliver them with high performance and suck it into this computer in order to spit out something new at a higher rate than maybe others can do, right? Because we want to be competitive. And to that end, we are leveraging a new European federation among the supercomputing centers, which is called the Lexis platform, and which we developed some years ago, which is now thriving. And this we will utilize, and the techniques from it we will utilize to build a big kind of data lake in order to ensure the high performance data access there.
You know, I was struck by something in Michal's keynote presentation where she had that idea of data over, it was a Venn diagram of data on one side and abstraction on the other side, and the intersection between the 2 points as where algorithms are being developed. One of the efforts that CLARA is trying to do is to really bring together multidisciplinary teams, not just from life science, but also from computer science. What are some other fields that maybe we might, given the fact that we're interested in these issues of more dynamic level systems and trying to think about how we might be able to pull other types of already established models into the CLARA framework that might give us, as somebody had mentioned, sort of a different viewpoint of how to look at the data that we've already collected. And that's actually a free question if anybody would like to take that.
It's okay. It's okay. And so I can say about between the disciplinary area, it is so important because in nowadays, because Europe and world is to face about this actual problem that now we have this so progressive implementation AI tools in the different areas. And so we understand IT for IT, it's, no, for me, and this is my opinion, it is not interest. But if we implement information technologies, tools, also artificial intelligence, models, methods, algorithms, etc., and in the special area, and where we can combine like this our knowledge, expert knowledge, and ethical knowledge is so important. And 3 points we need to combine in the special product. And so I and my department that I can present have experience in the a lot of different projects. And when we developed some special application or some programming models with AI, because it is our areas, and so we understand we also need to work together. And so collected data and data sets we get from our partners that made the request to collaboration with us. And I can say about the nice product with different pharmacy corporation like this Ukrainian Biopharma or Takeda, Japan. And with this organization like this immunologists in Ukraine, and we work together. And we can find this nice success when we combine our knowledge from different and multidisciplinary work.
Well, we can learn a lot from you on that in terms of, what kind of challenges do you see or expect it when trying to run these multidisciplinary teams in terms of sort of maintaining technical balance and really also sort of the human context of making sure that you've established a team? What have you seen that works well?
And the first of all, and we need to understand together, because we can see there's the same problem in the different side. And the first, we need to communicate a lot, explain, and discover what we need to find at the results. And so this is a big challenge. But when we find there is a combined solution, and so you can find the nice results and success of the product.
This is a question for everybody, but in terms of having new investigators, young investigators coming into your labs, are you finding them being almost inquisitive to ask questions when they don't know something, or perhaps maybe they're a little bit bashful or shy? And have you seen any trends over time? Are new researchers perhaps maybe a little bit more, aggressive and sort of wanting to know how things work, or maybe the opposite? I don't know.
Yeah, I have to say that, our new scientists and PhD students are, I would say, very active, not shy.
Excellent.
If they are shy, it's a little bit hard to do the science. You need to ask the questions. So, yeah, very active.
That's awesome. So, Josef, we've been working together for a long time, and we share a strong interest in the genetic factors of Alzheimer's disease. For those who don't know, we talked a little bit about the ApoE gene that comes in sort of 3 different flavors, E2, E3, and E4, and those variants really sort of provide almost 6 different versions of the ApoE protein that get produced. And each one of these really sort of can act as almost like a delivery truck for fat inside the brain. Some of those trucks don't operate so smoothly, and some like ApoE4, for example, can cause traffic jams that can increase and hasten the risk of Alzheimer's disease compared to others in life. And you and your team are building sort of these advanced computer models to simulate how these proteins interact and with fats in the brain to sort of see where that delivery process breaks down. Could you describe what makes the modeling so challenging and how new tools like what Stefana's building with the quantum and hybrid computing and others, our friends in Ostrava, are doing, and how do you think these interactions could be sort of more important in greater detail?
Yeah, thank you very much for this question. First, I want to say also interdisciplinary. It's sort of wonderful being part of this, interdisciplinary group of researchers. It is a hard work, to understand each other, but I think it's worth it. It's sort of very interesting. We learn from each other. The second point I also want to say, I would like to welcome our colleagues from Ukraine here, both on the stage, but also all the students here. It's wonderful to have you here. I would like to mention that so we have also close relation to the Ukraine Catholic University, which should be also here in Lviv. But welcome to both, of course, universities here. And I have several Ukrainian students in the team. I think I see here there's Anton and Roman down here. Please wave your. So come talk to us. We will be very happy to learn what you're working on and happy to tell you more about our work. And Anton and Roman are going to talk tomorrow. It's also an invitation. Are going to talk tomorrow in the Young Researcher Forum about some of their work. So back to the scientific questions. Yes, so we are working, and this is again, it's a joint work with the team of Jiří Damborský, who is not here today, but in Brno. But there are some representatives from his team also here. I think Sautuk Jimenez from his team. So it's a very much joint work. We work together. And it's so their team is specialized in structural molecular biology, and so we would like to really understand so these proteins APOE, they come in different variants, as I already mentioned, 2, 3, and 4, and they differ kind of on paper in very little, in just 2 amino acids. Yet they have this vast, downstream effect on the disease. So we would like to understand that better. Why is this happening? And the way we are doing it, we are modeling how these proteins, we can simulate that, how these proteins, how these different variants of the same protein, how they interact with each other, how they aggregate, but also how they interact with the other biomolecules, and especially we're focusing on lipids or the fats, because it's known that APOE is really responsible for transferring lipids in the body, but also in the brain. And so coming down to why it's difficult. So especially the interaction with the lipids are computationally very demanding. One can simulate those, but those are kind of very elongated, complicated molecules. So simulating them or the interaction with the protein takes a very long time. Sautuk can tell you we are talking days on supercomputers. And we are developing new sort of machine learning techniques which could speed up modeling these interactions, sort of orders of magnitude, ideally, so that we can sort of understand and model these interactions faster, get to the results faster.
Yeah, I think that's a really interesting point, and I'm sure there are people who are listening to this presentation, maybe in this room, that really sort of wonder about these time scales. And so certainly within your work, just the time scales of what some of these interactions look like at the cellular level, never mind the time scales involved in terms of running these simulations. Ondřej, I'd like to ask you a question in terms of, do your best here, but in terms of drug discovery, this is something that ultimately the public wants to know more about, and one of the questions that they're going to ask is, why is it it's seemingly such a slow process, and that there's trial and error involved, but your lab is really trying to change this picture, and like what can you say about how, for example, robotics and AI might be able to sort of speed this up and make it more efficient, and how does that workflow actually look in practice? I mean, if someone were to sort of be airdropped into your lab, what kinds of discoveries could they expect to be seeing?
Well, very good question. I would say you need to test almost every molecule in the lab. So the quantum computing, AI, you said speed up the process. So, and then you can selectively select the most valuable or important molecules and then test them in the lab. So that speed up the process. Another thing, it's and if we move to the humans, there are the regulations, very important regulations for safety and everything. That's another thing. How like the time it takes time, so that's the second more important thing. And yeah, you can speed up the process in the lab and then just test the more important molecules. Yeah.
Thank you. You know, it really sort of kind of helps stimulate the imagination a little bit. If you were to take sort of the person on the street into the laboratory, they might expect a lab full of microscopes and servers and data pipelines, but in terms of what we're trying to build here for CLARA and in terms of connecting those worlds, what has access to a digital infrastructure with AI tools and robotics and supercomputing allowed your team to do perhaps that maybe it wasn't able to do 5 or 10 years ago?
Question on me. What was the question again? Sorry.
So the sort of this expanded infrastructure of a CLARA where we are really sort of spread across a continent, where do you sort of see the opportunities to accelerate your work, and what sort of excites you about CLARA?
Sorry, I'm freeze right now.
Maybe I can jump in a little bit.
Yeah, just help me.
Because we are on the sort of AI part of this. I think what feels really exciting is that these sort of new, this is kind of one of also emerging trends, and I'm very happy that we are able to somehow capture that in CLARA already early in the writing, is that what's really emerging are these new sort of biological labs which are sort of automated. So it really enables like a new skill of experiments which are kind of, so we heard early in the presentations, we heard like the term agents and agents doing this, agents doing that. But I think what's really is beginning to emerge, and it's already emerging, that this sort of machine learning techniques or these agents, they can also actually design experiments and execute them in this maybe first semi-automatic, but eventually automatic manner. And I think the CLARA infrastructure, we are beginning to have also the AI infrastructure and the robotic sort of biological infrastructure to really join this together and have a sort of a new level of efficiency in both the, say, the virtual part of the screening, drug design, but also in the experimental execution, which is opening like a new scale where the machine learning is doing the virtual screening, but then also designing the experiment, what kind of mutations to test next in the lab, executing it, and then feeding this data back and training the model. And I think ultimately this really is going to speed up the current workflows, orders of magnitude.
Mm-hmm.
Yeah, Nataliia,
if I
you
can
want to jump in, please.
Yes, and I want to say about the growing infrastructure and using different tools that we need for our research and productive and development. And so the first, we need to be open also. And we need to use the source for representing your research and your data and your projects to also. And for example, Hugging Face platform. And so this is like the corporate, this is a nice idea for representing themselves. And also, we started and we opened from our department vast AI theories for using the different credits and training this model, because this is the first actual problem and this is a resource for training. This is a big, and in general, my colleagues understand, and we need to have the big source. And we ask it, and we made the request to our partners for support, because we are a university also. And so we have the nice big support from initiative, from my colleagues, from department, also PhD students. They are so motivated to grow our source and promotion. And also we use last year we have the open space like this AWS from also for support the partner. And I think in this area we need to collaborate and support each other, and that's so important. Partner, university, science departments. Thank you.
Okay, thank you. So we're going to sort of move a little bit into more of a thematic roundtable session here, a little mock roundtable session to perhaps maybe rethink the concept around brain resilience. This has sort of been an idea that we articulated in the CLARA proposal, and our overall mission is this idea that how can we maintain a neuron for 120 years? Ideally, that would sort of be an overarching aim, and perhaps by looking at how a neuron's metabolism operates, how its recycling and repair features operate, or how the signaling operates, and that we have basically brought together all the many disciplines under CLARA to sort of investigate these questions. But at a sort of more human level, you know, when you hear the phrase sustaining neuronal vitality, and this is just an open question, what does that mean to you personally as a scientist? Feel free to answer the question any way you like.
Okay.
Maybe we need wine.
No, I don't think I need wine. It's a difficult question because I would, since my background is not cellular biology, I think I would be tended to tackle it from a different perspective, which I don't think is better. It's just a different point of view. I would. Coming from AI and statistics, I think I would like to understand why the aging trajectories of different people are so different. And I think it's quite clear that we have now, with this huge real-life data, new opportunities to tackle that in a way that is quite different from traditional epidemiology. But doing that with real-life data also brings very different challenges, because in traditional epidemiology, you design a cohort, and by the way you design your cohort, you know that what you learn is not free of biases, but at least you can control them. When you try to go back in time from starting with the patient and going throughout their history, the design is really less well behaved. And so for me, there is a huge opportunity here in terms of understanding all the tiny factors that together we can use to prevent but in addition to treat brain disorders. So.
Yeah, no, I totally agree with you on that. And the way that, if I were to be asked the question, I almost think about this as really thinking about brain performance or individual performance, and how you can actually do the little things to sort of keep yourself going as long as possible. And I think that's perhaps one of the, it's a legacy of the 20th century, how we've done population level research in this sort of very prescribed way. But I think the reality is that given the computational power that we're getting, the idea of measuring more things and looking at more aspects of, how we operate will sort of allow us, for example, to see how our speech is staying the same or declining, or if we're getting enough sleep, or if perhaps our nutrition is not so well, and how that could ultimately impact cognition and or behavior or function. You know, that sort of gets to sort of this other idea where we were thinking about maybe going beyond the digital divide and starting to look at perhaps more analog-based signals. Stefan, I'm looking at you because you sort of operate in this space of multiple levels of data, but also multiple disciplines as well. And in terms of, as we think about the complexity of the brain, never mind the complexity of the person, you know, maybe perhaps we've had it all wrong. Maybe we need to do more real-time data collection to sort of track waveforms, maybe to look at blood flow in a different way, to actually look at a person, how they navigate a particular geographical space. And so all of that is sort of pointing to perhaps an explosion in more data collection. What are going to be some of the theoretical limits of being able to not only generate that data, but to process it, to archive it, and then ultimately to analyze it? And I'm thinking we're on scales of order higher than where we might be today.
Well, I think the answer to these, it was actually many questions in one, right?
It was.
So yeah, it's a bit, I will try to answer it in pieces, okay? So, I mean, one thing you can say always is, of course, we do more of the same, but that's not where we want to go qualitatively. Probably also we wouldn't have been given money for that. So, you can take two approaches to make a qualitative jump and not just quantity. And the one is certainly the automation approach, yeah, that you are able to collect more data more automatically with workflows that are automatized from the experiment down to the computation, then also inside the computing system or across the computing systems, which again was one of the aims that we have with the Lexis platform, to orchestrate computational workflows across different machines at the European computing centers. Now, this automation will enable a much higher throughput and a data collection at much other scales than before, and also the data processing at higher scales than before. And it will allow, for example, a PhD candidate not spend 50, 60, 70% of their time with actually launching some analysis and then digging into it manually and so on, but they will have 10,000 analyses that are all homogeneously done and delivered in a certain relatively short time, so they can work on much more data again. This is certainly something that goes all the way through, and we see this being taken up by the scientists, because we've been talking about workflow engines for 20, 30 years probably, but now we see that we have users doing it and actually users asking for it. For example, Nextflow, Galaxy, workflow orchestrators, Airflow, and so on. So people are starting to really leverage it. And the other thing is that, of course, in the computing itself, you can do qualitative changes, going from the classical digital computer to quantum computing, or we have also at LRC now a so-called digital analog quantum system, which is kind of photonic computing. So you can try different approaches to go away from this classical computing paradigm, or also from GPU computing paradigm, and go beyond and try to see where quantum computing accelerates your stuff or allows you to model, for example, waveforms or whatever in a better way. I'm not a quantum computing expert, I must say, but certainly, for example, encryption goes super fast, certain other modelings goes very fast, certain searches go very fast, so that we see as a very potent accelerator for a number of tasks that will certainly be profited here and also in the CLARA. Yeah. We just have to know and understand where we can really leverage it the best way. And that's, I think, a challenge which I'm really happy that in this center we're tackling it basically as the co-evolution with science, so that you as a scientist can be at the forefront and we at the computing centers at the same time, basically. Yeah.
Thank you. That was a really good answer.
A bit lengthy, sorry. Yeah.
That's okay. That was perfect. Josef, you know, I'm curious. You know, we're always trying to find new talent. If you were to find a curious teenager, how would you describe their work to them, and how would you use that description to maybe try to recruit them to, you know, find their way to your lab at some point?
Okay, it's an interesting question. Actually, I'll use the opportunity to answer one of the previous questions, like about the vitality of the cell and of the neuron. And it's so I've spent, my background is in computer vision and robotics, so understanding images, videos. But for about like last, I would say 5 or 6 years, I had the wonderful opportunity to collaborate with excellent protein engineers on molecular biology. And it really took us like, at least me, several years before I understood and developed the vocabulary, intuitions in the domains, and it really, talking to experts all the time to understand it. So I'm at this level. After these 5 years, I've sort of developed some intuition. Of course, I'm not a biology expert, but I've developed some intuitions for the proteins, small molecules, proteins like a chain of beads, which folds into a certain structure, which is dynamic and moves, and then this relates to the function of the protein. You can think of it as an engine which opens and closes, and does different things across the body. But now, sort of coming to the cell, to the neuron. So just thinking of the scale of this. So the protein, if you come to the cell, it's almost like this little guy coming to the building. Yeah. So it's and of course there's a receptor maybe as a door which allows you to enter, but it is like a hugely vast difference in scales. And of course we come about to have tools where we can model on the level of one protein or its interactions. But now we are in front of this building, which is the cell, and there is this area called cellular biology, which I read a paper, try to read a paper, I don't understand anything suddenly. So even in biology, suddenly there is like a whole, for me it was a very sort of humbling experience coming now to the cell, with this protein as a protein in front of this, door, this receptor to the building, and trying to, so how do we model this cell, and how do we model the processes inside the cell? Of course, this whole area, but also how do we measure? Because we like to understand the effects of if you have a different protein on a variation coming to the door, how is it going to interact differently with the door, for example, and how is it going to affect what's happening inside this building? So I think these are some of the very interesting questions. Really great to understand and have help of the our interested teenagers to address.
Yeah, I was reminded, I think at the first meeting here in Prague, I was asked a question by this young man, and I was so blown away by the level of precision and detail. And then I asked him, so what year are you in your PhD program? He said, oh, no, I'm finishing up high school. So I'm going to just take a pause and see if there's a question. I think Adam has a question, and Ira has a question, and maybe I could take your microphone.
So my question probably actually was stimulated by your very first question you asked about the fact that there's a heck of a lot we don't understand about the disease biology at a fundamental level, and then the notion that what's it going to take to have a neuron, hopefully more than one, last for 120 years. So that sort of brought to mind the recent work that was done, I think, in the Netherlands, where they looked at these super ages. So these are people that have lived very healthily, great cognitive function to 100 years old or so. And they tried to figure out what was it about the biology of these people that might explain that. And as I recall, I'm not sort of super expert in that study, but as I recall, it was sort of a combination of things that they probably wouldn't have suspected ahead of time in terms of biological signals, biomarkers, analytes in the CSF, and so forth. So I was just thinking that I wondered what the panel might think about the idea of using that as, in a way, it's sort of a biologically validated set of targets or ways into the biology, and using those as, because today we've been talking a lot on the abstract,
right?
About biology, about massive computational resources and so forth. But here we have I don't know how many there were, maybe 9 or 12 specific markers, which presumably we know or can find out what they do biologically, and using those as inputs to some of the things that the project is trying to tackle. So that was brought to mind when you asked your first question, and I just wondered what the panel thought about using that relatively recent discovery as a, kind of a way into the biology.
I think it's a great question, obviously, and it certainly represents an opportunity to almost have like a perfect control. I think there's a number of these types of populations where you have the super agers that you can actually maybe start to do that, and maybe to perhaps sample a whole host of various fluid markers and other types of markers, maybe to sort of establish that panel. I think that really also speaks to something. I think, Ira, you were talking a little bit about having more heterogeneous sampling frames, sort of beyond just what we have of Central Europeans and Northern Europeans in a lot of these samples. And because I think these there's different populations within the world that sort of represent this, and then maybe that would be sort of a great opportunity to sort of mine for that kind of information. I think the issue also then becomes, in terms of how do you sustain that as almost like a living laboratory, the issue of a Framingham-like study that could be sort of conducted as a population-level observatory. It's nice, but from my perspective, I don't think that governments and society are willing to sustain that. So that sort of comes back a little bit more to something that Olivier really sort of brings to mind, which is how can we start to use more real-world evidence to sort of develop that? And I think there is an opportunity to start developing sort of a dual use between research and clinical medicine, in the sense that there shouldn't be this divide, that it actually needs to be fused at one level. And then I think at another level, we also need to sort of think about how we're going to use and deploy technology to get further into populations, to get into sort of that last kilometer, so that we really are now no longer sampling just unique groups, but perhaps much larger populations, and sort of mining that data and while delivering care at the same time. So I think that's where the opportunity is, but I believe that might be a way to get to it. I don't know if anybody else has some other ideas.
Mechanistically as well, not necessarily a large data collection exercise or epidemiological study, but if we know these analytes, then mechanistically what do they do? And then if you're thinking about ways into modeling the biology, then they might give some clues. That was where I was going.
Maybe just answer then, like from again, we are on this APOE protein and modeling that, but I think there are some of these rare mutations like the Christchurch, which we kind of know can measure the effect. But we can we talked about the 3-2-3-4, but actually we can also simulate. You can do the same analysis for these rare mutations to understand through mechanistically their effect. And of course we are also doing that there. So it's a great point.
And then one last comment, I don't think we got a chance to really talk about it today too much, but hopefully next year we'll be able to go a little bit deeper into proteomics and transcriptomics, because I think that's going to be sort of the next pathway. And I know that Ira's group is very much interested in this, and they've done a wonderful job of not only sort of setting the stage with the clinical level data, but also that fluid marker data. Ira?
Yeah, first of all, I wanted to invite Josef to look into our data, because interestingly, in Italy, we see the highest prevalence of dementia in Europe, but they have relatively low ApoE expression for expression, in opposite to us in Northern Europe, where we have in our cohort 45% expressed ApoE4 allele, but we have a lower prevalence of dementia. So we are back to this modeling, and which factors are compensating negative genetic pressure. That was the one thing, and the other is to you talked about the supercomputer interacting or being federated soon, Stefan. But Jülich, for example, can't handle highly sensitive data. I don't know if Leipzig can do so. I think this will also be complicated. So what are you doing from the research perspective? What are your efforts to make your resources available for real-world data of significance?
Yeah, so we see this problem with the sensitive data as well, and in particular on the HPC biggest clusters, because these clusters have been constructed from a physics and engineering background where data protection plays no role whatsoever, except for maybe you don't want to know, you don't want to let out your plans on the newest bomb to the enemy, but in fact, lots of it is from bomb simulations, right? Yeah? And that have been then, the privacy has been guaranteed by putting the computer to the lab, often to a national lab, for example, in the US. Yeah, so, okay, so coming from that story, we have now, for example, in LRC a secure cloud that we are developing. So that's still a prototype, and it's a cloud infrastructure. It's not a high-performance computing infrastructure. And there is like all the way through the encryption from the file system to the virtual machines. And that is our tries to get into this medical data elaboration. And I think from these infrastructures we can then learn for bigger systems. So from these very first fully encrypted systems, we have to then transfer the methods and the privacy keeping methods to the bigger clusters. And then in the end we will be with a mixed system landscape that would hopefully serve most of the researchers. I would hope that these smaller clouds can already serve maybe 90% of the researchers that work with small data, but then we need also to answer the question for the high performance computing and the AI big systems and so on, which is in this project. And so the first approach is to split the problem and begin to attack it from two sides, from the small, very secure side and from the big, less secure side, and then to somehow merge these approaches together.
Okay, well, there it is. Well, I want to thank the panel for spending this hour with me and answering these questions. And please give them a big hand.